यूट्यूब में स्ट्रीम में या गूगल मीट में? अच्छा, मैं कर रही हूँ सर। वो पता नहीं स्ट्रीम हमने टेस्ट किया था पहले, और अब स्ट्रीम नहीं हो पा रहा है, पता नहीं क्या करना। और टेस्ट किया था, हाँ भी स्ट्रीम के लिए दिन है 12:45 हो गया है, फिर भी स्ट्रीम हो रहा है, होने लगा। ठीक है सर। गूगल गूगल मीट का मैं करती हूँ। अच्छा ठीक है, ठीक है सर। चलो 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 यार,
नहीं मैं खाना नहीं।
कर लें स्टार्ट।
With your permission, sir.
अमनतुल्ला, हाँ ठीक है।
Yes.
सर अब स्टार्ट करते हैं।
कुछ इको आ रहा है।
कैसे? सर ये डॉक्टर अमनतुल्ला है, हमारे स्पीकर हैं आज के। और अमनतुल्ला सर हमारे प्रिंसिपल सर हैं। ये तो कहाँ से खोलूं? स्टार्ट करने के लिए मैं शुरू कर रही हूँ टाइम।
हेलो।
हाँ अमनतुल्ला।
मीनाक्षी।
हाँ अमनतुल्ला, स्टार्ट कर रहे हैं।
चलो।
कोई प्रॉब्लम है? आवाज आ रहा है ना?
इको आ रहा है, इको।
इको आ रहा है।
इको। So please, whoever is not speaking, please put yourself on mute.
निशिकांत सर,
हाँ मैडम।
ये सर वाला जो है ना उसको म्यूट कर दीजिए तो।
सर वाला एक्टिव कर दीजिए मैडम। सर का है ना लैपटॉप है बैठे हुए, उनका एक्टिव कर दीजिए।
नहीं, उनका एक्टिव है।
जिस अच्छा, जब क्वेश्चन पूछना हो तो हम अनम्यूट करें, अदरवाइज म्यूट कर दे।
ठीक है, ठीक है,
नहीं तो एक इको होगा।
ठीक है। सुनो ना, अरे सुनो, ये प्रिंसिपल सर का म्यूट कर सकती हूँ मैं? कैसे?
हेलो हेलो।
कैसे? निशिका, ये वाला म्यूट करो।
मीनाक्षी।
अब ठीक है, चलो।
उसका तुरंत अनम्यूट भी कर देना।
तुम बाहर निकलो ना जल्दी, यहीं बैठो ना। अब ठीक है अमनतुल्ला? अब ठीक है?
हाँ तो स्टार्ट करें।
हाँ स्टार्ट कर रहे हैं। वी आर स्टार्टिंग। सर, प्रिंसिपल सर, शैल आई स्टार्ट? Uh, a very warm good afternoon to all. Respected Dr. Nityanand Prasad, chairperson of the event, and our principal, respected senior colleagues, friends, and above all, dear students. On the behalf of Government Polytechnic Gaya, I, Dr. Minakshi, lecturer chemistry and convener of the event, would like to welcome all the distinguished participants and especially our guest and speaker of the day, and also a very dear friend of mine, Dr. Amantullah, to this wonderful webinar on a very interesting and pertinent topic, COVID 19, a new challenge for the humanity. We are all aware of COVID 19 pandemic and the set of challenges it has brought with it. The world has changed completely. Our working culture, our priorities, how do we meet and greet each other has changed completely. There are umpteen numbers of example on the things that has been affected and has been in the popular discourse, as well as in the very many topics of the numerous webinars that has suddenly become the flavor of the past few months. Lately, we have been hearing a lot about coronavirus and also about the possible treatments, that is, vaccines, several antiviral drugs, several formulations, preventive measures, etc. But do we really have the insight of COVID 19 pandemic and the steps that are being undertaken to fight against the disease through designing and development of drugs? Do we really know why it is proving so difficult to make a drug or vaccine against this pandemic? What are the steps in the drug development? What are clinical trials? A word that becomes so common today. To give and to further enhance our understanding about this novel disease, we are very fortunate that today we have amongst us Dr. Amantullah, a scientist par excellence. But before going on to him, I would request our principal sir and chairperson Dr. Nityanand to say a few words and formally welcome our speaker.
Sir, you are muted. Please unmute yourself.
Uh, यार इसको करो ना मुझे वहां से बोलो ना इसको पकड़ने के लिए अरे इसको अनम्यूट करो ना प्लीज आकर पहले अनम्यूट करो निशिकांत सर को अनम्यूट करिए ना निशिकांत सर
Thank you, Dr. Minakshi. Also, audience, audience, सुनाई पड़ रहा है?
Yes.
Yes. Thank you, Dr. Minakshi. As Dr. Minakshi has mentioned, how COVID-19 pandemic has affected us. One thing I would like to stress upon to this coronavirus is the effect of COVID-19 on the education system. The education system is experiencing a fundamental change, and we ourselves are experiencing it. The traditional talk and talk method of teaching has suddenly went to online mode of teaching, and now it is becoming more clear that this
सुनो ना
online mode
वो
of
आजकल
teaching
प्रेजेंट करने लगा अपने आप को इनको ये भी नहीं पता कि बोलते कैसे इसको रोकते कैसे हैं हाँ बंद है
is only mode of service
राज
address
कंपनी
the
से मत
one
म्यूट करना इस पर।
and will
राज
speak
कंपनी
from the
के बाद
teacher
प्रेजेंट।
to those who are being taught, that is, the learners or students. Now your source of information on a particular topic is not only the teacher in the class, but also the different types of learning sources available to you online. The role of now who will guide you through the sea of information, but
हाँ सर।
what you want to pick up is left entirely upon you.
क्या?
So, my dear students, you need to buckle up and need to be very attentive to the guidance that is being provided to you by your respected faculty members. There has been a lot of talk about the COVID-19 pandemic, but as it is said, there are two sides of it. This COVID-19 has brought us some opportunities also. One of the opportunities is this webinar. It is of great pleasure that today we have amongst us a distinguished talk on COVID-19. On any other day before this pandemic, it would have been difficult to think of such a setting in such a short notice and with such minimal requirements. As chairperson of the event and also the principal and head of the government of Technical, I welcome Dr. Amanpura and extend my warm wishes for his invitation, and I am sure it will be about the thank you.
Thank you, sir. Meenakshi. Hello, Meenakshi.
यस यस।
ये प्रोजेक्ट नहीं हो रहा है।
हाँ
प्रोजेक्ट नहीं हो रहा है।
प्रोजेक्ट नहीं हो रहा है। अमनतुल्ला प्रेजेंट करो।
ये दिख रहा है स्क्रीन?
हाँ हाँ अब दिखने लगा अमनतुल्ला। थैंक यू प्रिंसिपल सर।
Thank you.
Thank you, Principal Sir, for your insightful observation on effect of COVID-19 pandemic on educational system and for welcoming our speaker of the day. Dear participants, before I present the mic to Dr. Amantullah for his presentation, I would like to take this opportunity to share some information about him. Dr. Amantullah did his MSc Chemistry from Banaras Hindu University in 2005 and was a gold medalist in his subject. After that, he qualified CSIR JRF in 2005 itself and GATE in 2006 and secured All India Rank 2. After that, to pursue his doctoral work, he joined CSIR's Central Drug Research Institute, Lucknow, one of the premier research laboratories in India, and has his PhD registration from BHU. During his PhD days, he impressed everybody in the institute, be it his peers or students or the technical staff. His oral presentation during the CDRI Diamond Jubilee Celebration Series lecture was adjudged as the best oral presentation in chemical sciences. His PhD thesis won the first prize in Ali Lilly Asia Outstanding Thesis Award in 2011. After PhD, he went on to join the pharmaceutical industry and joined Integral Biosciences Noida, which is a company focused on to join the pharmaceutical which is a company focused on drug research and development as research scientist, and presently he is a principal scientist. In simpler terms, he joined as a team member, went to become a team leader, and now he manages a network of teams. He has published his research paper work in international journals of repute, among the best in the world, such as journals from Royal Society of Chemistry and American Chemical Society. He also has four international patents. Though he works in the industry, but he's basically an academic person, a teacher at heart. His work in the industry has not deterred his craving for teaching and learning. He has his own YouTube channel on chemistry subject by the name ChemWiz, where he posts his lectures on a range of topics in chemical sciences. Finally, I would like to highlight few things about his persona is that of all the success he was achieved in his life, he is still a very down to earth, kind, generous, supportive and honest person, committed to his work and above all always has a smile on his face. Now I would request Dr. Amantullah to present his lecture.
So is everybody able to see the screen? Means my presentation.
नहीं
Hello?
नहीं दिख रहा है अमंतुल्लाह, नहीं शो हो रहा है।
Oh,
दिख रहा है सर बोल रहे हैं
थोड़ा देर
सा
पहले
दिख रहा
ट्राई
है।
किया था। ये presentation दिखा है। ये-
Just your open presentation, sir. Everything is okay.
I have opened the presentation.
Okay.
Is it visible now?
Not visible.
Let me recorrect.
Not visible. हाँ, अब आएगा हट। हटो।
Now?
Now it is visible. Now we can see. Yes.
Okay.
Okay.
So should I start?
Yes.
So good afternoon, everyone, and thank you, Dr. Meenakshi, for the nice introduction. Thank you very much, the organizing team, for organizing this webinar. Hope you all are doing well, staying at home, following the instruction for your safety and for the safety of others. The topic of the webinar is COVID-19, a new challenge for the humanity. In this, we will be discussing the following points: introduction, case fatality, development of pneumonia-like symptom in COVID-19, pathogen. Under this head, we will be discussing about the structure of the pathogen, pathogenesis phases, multiplication, transmission, and the next heading will be diagnosis of COVID-19, then COVID-19 treatment, COVID-19 prevention, and finally we'll draw some conclusion. So let's start with the introduction. COVID-19, which is the short form of coronavirus disease 2019, it's a very contagious pneumonia-like respiratory disease caused by severe acute respiratory syndrome coronavirus 2, which is abbreviated as SARS-CoV-2. Initially, WHO called this virus as novel coronavirus. Affected organs. Lungs are the most affected organs in COVID-19. Apart from lung, heart, kidneys, brain, intestine, and blood vessels can also get affected. In December 2019, pneumonia of unknown cause occurred in Wuhan, Hubei province of China. On 7th of Jan 2020, a novel coronavirus called SARS-CoV-2 was identified from the sample of patient. WHO announced this epidemic disease caused by SARS-CoV-2 as COVID-19. On 31st Jan 2020, WHO declared a global health emergency, and subsequently, on 11th of March 2020, WHO declared it as a pandemic. Since then, it's continuously increasing day by day. Globally, if you can see, there are 1,55,81,009 confirmed cases so far, with 6,35,173 deaths. If you see in India, there are 30,36,861 confirmed cases, with 31,358 deaths. This data is one day before. So we will be reaching around 14 lakhs as far as the confirmed cases is concerned. Now the case fatality. The fatality of COVID-19 depends on three factors: age, pre-existing disease condition, and gender. This graph shows COVID-19 mortality rate by age. Here we can see that the mortality rate was almost zero in the patient with the age ranging from 0 to 9 years, but in the patient with the age ranging from 0 to 39 years, it is around 0.2%. And subsequently, it increases with the increase in the age. Here we can see that the patients with 80 plus years age are more prone towards the mortality of the COVID-19. Means the mortality rate is around 15%. This graph shows death rate of COVID-19 patients with pre-existing disease condition. Here we can see that the patients with no pre-existing disease condition are less mortal, means the mortality rate is very low, with around 1%. But in the patients with the pre-existing disease condition like cancer, high blood pressure, chronic respiratory disease, diabetes, and heart diseases, their mortality rate is higher. It is 5.6%, 6%, 6.3%, 7.3%, and 10.5% respectively. The mortality rate also depends on gender. There are growing number of evidences that males are more fatal for COVID-19 than the female. There is a scientific reason that we can discuss later. Development of pneumonia symptom in COVID-19. This is the respiratory tract of a human body. Normally, when we breathe, air moves through the trachea, the windpipe, and through the large tube called bronchi, and then through the smaller tubes called bronchioles, and finally into small sacs called alveoli. In alveoli, there is exchange between oxygen and carbon dioxide. In a healthy person, the immunity system is very strong. So, when any pathogen like virus, bacteria, etc., when enter into the lung, our immune cell attacks on the pathogen like this and kill them. Then they are subsequently removed in the sputum, means in mucus, by coughing. But in the case when our immunity system is very down, like in the case of COVID-19, then the virus overwhelms the immune cell and our immune cell is not able to kill the virus. So, because of this, a large number of chemicals are secreted in response for the immune response, and that chemicals cause a severe inflammation in bronchioles and alveoli. Because of that, there is accumulation of a large number of fluid in alveoli, which interfere with our body to take oxygen. So this is the condition of pneumonia. You can develop a pneumonia-like condition only in one lung, which is called lobar pneumonia, or many parts of both the lobes can be affected. This is called bronchopneumonia. Pathogen, as we have already seen that the pathogen for this COVID disease is severe acute respiratory syndrome coronavirus 2, which is abbreviated as SARS-CoV-2. This is the third virus from the coronavirus family. The first one is severe acute respiratory syndrome SARS that was found in China during 2002 to 2003 that has caused pandemic. And second one is Middle East respiratory syndrome MERS that originated in Arabian Peninsula in 2012. Now we will discuss some general thing about viruses. So as you might be knowing already, the viruses are intermediate between living and non-living. They can lie outside the living system just like a dust particle, but as soon as they enter into the living system like cell, they start multiplying, which is the characteristic of a living being. The viruses consist of two parts. One is nucleic acid and other is proteins. The nucleic acid can be either RNA or DNA, but not both at a time. Structure of SARS-CoV-2. The SARS-CoV-2 virus appears as a ball with several spikes on a surface. The diameter of SARS-CoV-2 is 60 to 140 nanometer. The genetic material is single-stranded RNA coated with a protein called nuclear protein. The whole genetic material is enclosed in an envelope consisting of a plasma membrane, that is a lipid membrane, with envelope proteins and membrane proteins. From the envelope, several spikes emanate that are called spike proteins, and these spike proteins give this SARS-CoV-2 as the appearance of a crown. So because of that, because of the appearance just like a crown, it is named as coronavirus, because in Latin, corona means crown. Pathogenesis phases, or the infection phases of coronavirus 2. There are three phases. The first one is incubation period. Actually, incubation period of a pathogen is the period between the exposure to the pathogen and appearance of the symptom. In coronavirus, on an average, this phase is of 5 to 6 days, but in some cases, it can be less than 5, or in other cases, it can be as long as 14 days. The incubation period is followed by second phase, which is emergence of symptoms. The common symptoms are fever, cough, fatigue, sore throat, aches, shortness of breath, diarrhea, anosmia, which is also called loss of smell, ageusia, called loss of taste. In around 90 to 95% positive cases, the disease is self-limiting, and in 25 to 50% positive cases, it is asymptomatic, when means the patient doesn't develop any symptom at all. The third phase is severe disease. In this phase, only 5 to 10% patients enter. This is characterized by bilateral pneumonia that I have discussed earlier and oxygen desaturation. These two symptoms appear within 2 weeks after the infection. Around 2.5% patients enter into the acute ARDS phase, which is failure of the respiratory system, and some of the patients have multi-organ failure. SARS-CoV-2 replication. In this, we will learn how the SARS-CoV-2 multiplies within the host cell. This is the part of it. This is the structural part of a living cell. The virus is lying outside the cell. For the multiplication to occur, the virus should enter inside the cell. For this, there is a protein on the cell surface called angiotensin converting enzyme 2, S2. And with S2, the spike protein of virus binds like this. After binding between the spike protein A and S2, there is change in the conformation of this membrane that leads to endocytosis, by which the virus enters into the cytosol in a small sac called endosome. Then this endosome fuses with a large sac called lysosome. Lysosome have several digestive enzymes, and after digestion, the viral RNA comes out to the cytosol, where it recruits ribosomes. You know, ribosomes are responsible for the protein synthesis. So, with the help of ribosome, a large number of viral proteins are synthesized. Among them, one of the proteins is RDRP. RDRP means RNA. This is RNA polymerase enzyme. Means RNA dependent RNA polymerase. The full form of this is the full form of RDRP. This RDRP then synthesizes several viral RNA. All these processes, means synthesis of protein and this synthesis of RNA, occurs inside the endoplasmic reticulum. Now these things enter into the Golgi body, where RNA and protein combine with each other to produce progeny viruses. These viruses can go out by exocytosis, and then they will infect cells in the vicinity. So this is in short the SARS-CoV replication. Transmission of SARS-CoV-2. In this, we will learn how the COVID virus is transferred from one person to another. So there are two modes. One is direct mode and another is indirect mode. A COVID positive person expels out droplets consisting of a large number of coronaviruses while exhalation, sneezing, or coughing. If that person has another person in his vicinity, these droplets can enter into his respiratory tract via inhalation, and that will cause the infection to that person. So this is the example of direct mode of transmission. Viruses can survive in the air for 2 to 3 hours. So these droplets may drop on certain objects like TV, telephone, bench, etc. So if a person touches this object and subsequently he touches his or her mouth or nose, then the virus will get into his or her respiratory tract and the person will get infected. So this mode is indirect mode. There are evidence that the virus can be transferred from human to human. It can also be transferred from human to pets, but there is no concrete evidence that can support pet to human transmission. COVID diagnosis. If you have corona-like symptom, as we have discussed earlier, so first of all, you consult your doctor or call the helpline number. Tell your doctor about your symptoms, about your travel history, or about if you have contacted any COVID positive patient. Based on the counseling, your doctor will decide whether or not you have to go for COVID test. If your doctor recommends for COVID test, then you will go for the COVID test where the doctor can take sample from your nose or from your mouth with the help of long swab. Then the sample will be sent for the diagnosis to the diagnosis center. The WHO has recommended 2 types of tests. One is molecular test, which is also called RT-PCR test. In this test, viral RNA is directly detected. It is a very accurate test, but it is costly and time-consuming. The second test is antigen test. In this, the viral proteins are detected. It's less accurate, less costly, and rapid. There is one more test that WHO doesn't recommend, which is antibody test. It is basically a blood test that detects antibody produced by your body to fight against the coronavirus. So it detects past infection, but it cannot detect current infection. It is less costly, but rapid. COVID treatment. Currently, there is no COVID treatment available so far. Efforts are going on towards the development of vaccines and antiviral drugs. So we can divide efforts into 2 parts. Efforts towards making vaccine and efforts towards the development of antiviral drugs. So the vaccines are the inactive form of the pathogen or its body part that are injected into the human. After injection, this triggers our immunity cell to synthesize antibody against that pathogen. Currently, there are 23 vaccines in clinical trials. Clinical trials means they are being tested in human. And apart from these 23, there are 140 in preclinical trials, means they are being tested in non-human, like mouse, rat, or guinea pig. The vaccine from Sinovac, University of Oxford and AstraZeneca, and Moderna are most promising. There are 2 Indian companies too, like Bharat Biotech and Zydus Cadila. Their vaccines also have entered into clinic. But the vaccines from the first 3 companies have crossed the phase 2 clinical trials, and currently these are under phase 3 clinical trials. But the vaccine from Bharat Biotech and Zydus Cadila, these are just in phase 1, phase 2 clinical trials. Now let's talk about antiviral drugs. So there is no new antiviral drugs. The people are trying it pre-existing antiviral drugs that have shown some efficacy against COVID. Among these, the three drugs are hydroxychloroquine, favipiravir, and remdesivir. So let's discuss about these three drugs and their mode of action one by one. Hydroxychloroquine. Basically, it's an anti-malarial drug. The structure of hydroxychloroquine is like this. Let's represent this hydroxychloroquine with a star. And now recall how the viruses multiply inside the cell. We have just discussed. So on the cell surface, there is one more receptor near S2 called sialic acid. This is acidic in nature, and our hydroxychloroquine is basic in nature. So there will be an acid-base interaction between hydroxychloroquine and sialic acid like this. So this binding of hydroxychloroquine with sialic acid would inhibit the binding of virus with S2. And thus, it will inhibit the entry of virus into the cytosol. It has also been found that this hydroxychloroquine acts as an ionophore and increase the concentration of zinc ion inside the cytosol. This increased concentration of zinc ion inhibits this RDRP protein. As a result, the synthesis of viral RNA is inhibited, and because of that, the viral replication is inhibited. There is third thing. This is, as we know that HCQ is basic, it enters into the lysosome. After entering, it makes the pH of lysosome higher. With the higher pH, it is not able to join with this endosome. So we can see here that this hydroxychloroquine is working with three different ways to restrict this virus from the multiplication. The second drug is favipiravir. This is the drug for influenza. It was developed by a Japanese company. This is the structure of favipiravir. It enters the cell, and after its entry, it is converted into favipiravir ribose triphosphate. This favipiravir ribose triphosphate mimics adenosine triphosphate and thus inhibits this RDRP. So once RDRP is inhibited, there is no synthesis of viral RNA and there will be no formation of progeny viruses. The third drug is remdesivir. This is the drug developed for Ebola virus. It was developed by an American pharmaceutical company called Gilead in 2009. This is the structure of remdesivir. It enters the cell in the form of GS441524. This is just a number, it's compound number from the Gilead. This is converted into GS441524, and then GS441524 after triphosphorylation is converted into GS441524 triphosphate. This also mimics adenosine triphosphate and thus inhibit RDRP. So once RDRP is inhibited, the synthesis of viral RNA is inhibited, that ultimately inhibits the viral replication. COVID-19 prevention. Since there is no effective drug or vaccine for coronavirus till date, so we will have to be dependent on only prevention. These are clean your hands, often using soap and water or alcohol-based sanitizer. Maintain a safe distance from anyone who is coughing or sneezing. Wear a mask properly. Don't touch your eyes, nose, or mouth. Cover your nose and mouth with your bent elbow or tissue while coughing or sneezing. Stay home if you feel unwell. If you have a fever, cough, or difficulty in breathing, seek medications. Now, conclusion. COVID-19 is a serious global public health issue. It has badly affected our economy by sharp rise in unemployment, stress on supply chains, decrease in government income, collapse of tourism industry, reduced consumer activity, and collapse of hospitality industry. There is unmet need for COVID-19 vaccine or drug to save human beings. Hopefully, we will get vaccine by the end of this year in India. With this, thank you very much for listening.
Thank you very much, Amantullah. Now I would like to call upon Ramu sir, our convener. Please, Ramu sir.
Good afternoon to all. I am Prasad, co-convener of the webinar committee. On behalf of the Government Polytechnic Gaya, Principal Sir and faculty members, extend a very hearty vote of thanks to all the distinguished participants who blessed us with their presence and took out valuable time of their busy schedule, both on Google Meet platforms as well as on YouTube stream. First and foremost, I thank our guest and speaker of the day, Dr. Amantullah, who spared his precious time for us and graced this occasion with his presence and delivered such insightful lecture on COVID-19. Sir, your lecture helped us to know COVID-19 in a more scientific way. It was indeed a very intriguing session where we came to know many different aspects of COVID-19. A warm thanks go to our principal sir for his vision and commitment to work and its execution. Behind every successful program lies the manifold attempts of our committed faculty. I would like to take a moment to acknowledge the immense contribution of our dedicated faculty. At the last, I would like to extend our special thanks to all the students who attended this lecture. Thank you all for making this program a huge success. With the permission of the chair, I will now call upon the session to end. Thank you again for your participation.
Thank you very much, sir. Thank you, Amantullah. Thank you very much.
Thank you, ma'am.
Thank you, everybody, for joining.
चलिए तुम बोले सुना दिया कसम